Okay, so I get to be the lucky one who introduces Elli. So I'm Kim Nye. I hope you guys are all getting to know me at this point. President and founder of TESS Research Foundation, where our goal is to help all of you families who have been diagnosed with this disorder and to make as much progress as we can towards treatments and cures. And the very beginning of that journey includes everyone receiving a diagnosis. And so today we're going to talk about what a diagnosis looks like. We'll look at some of the paperwork. And the master of ceremonies, the one leading the presentation, is Elli Brimble. And Elli is the head of clinical operations at Citizen. She is also a certified genetic counselor by training, and before her job at Citizen, she worked at Stanford Children's Hospital. I think, Elli, I'll just hand it over to you and you can introduce yourself further if there's anything that you want to add before your presentation. But we're so grateful to have you here today and we can't wait to see your presentation.
Yeah, it's such a pleasure to be speaking with this group. It's a special group near and dear to my heart, so I'm grateful for the opportunity. Can everyone see my slides okay? Okay, great. Excuse me. So today I really wanted to start off by sharing some of the foundational knowledge about genetics that helps guide our understanding of inherited conditions. So my expertise is really in the genetics of neurologic disorders. If you have any questions that are maybe a little bit more specific to SLC13A5 deficiency that I'm not able to answer today, no problem. Ask the question anyways, and we will find an answer for you shortly, if not on this presentation. So to get started, I'm going to introduce some terms that are commonly used in describing genetic conditions. And this way we have a common vocabulary to really lay the framework for our discussion today. So starting off with the basics, when we hear the word gene, it's really referencing a single piece of DNA that codes for an instruction in the body. You might hear there's this sort of one gene, one protein rule. It's not a hard and fast rule, it changes sometime, but we can always think about one gene as resulting in an instruction. That instruction or that gene can be thought of as a string of chemical letters. And there are really only 4 letters that are ever used to make up a genetic instruction in the body, and you can see them here, A, G, T, and C. What's interesting for the genetics of health conditions is that the sequence these letters occur in the genetic instruction is really important to ensure that the instruction remains accurate. We know that changes to the spelling of our genetic instructions might impact the way that gene works. Most genes actually store the instruction for a protein. And so this is really what this image is showing you. It's showing DNA, and we can see very small here the letters A, T, G, and C. And this is actually being turned into a protein. So the genetic instruction codes for a protein. And proteins are really just molecules that perform important jobs in our body. So some proteins make chemical reactions, other proteins act as signals, other proteins form structures in the cell. And so there are really many, many different functions that our proteins are doing. So to really, break it down and summarize, our genes are the blueprints carrying an instruction for a protein. And the proteins are the things that are actually doing jobs in our body to help it function, develop, grow the way it's supposed to. There are more than 20,000 characterized proteins in the human body. That characterized word just means that we have some understanding that they exist and what they do. And so I'm sure there are many more uncharacterized proteins as well that we have yet to learn about in detail. And what may be a little bit surprising is that most of these proteins are actually expressed in the human brain, meaning that they are turned on and doing some job in the brain. That is a lot, but when we think about the natural complexity of the human brain, it's maybe not so surprising that there are many, many jobs that need to be done in order for the brain to work properly. Some human conditions are caused by changes to the spelling of our genes. So this example here is a fake sequence of letters, but we can imagine that it's a gene. And again, you're really only seeing the letters A, T, G, and C, and they're in a specific order. And what I'm highlighting here is that you can see a letter C has been changed into a letter G. It's a very, very small change, one that we would call a variant or a mutation. But depending on where that change occurs and what gene it occurs in, it can have a really big impact and potentially cause symptoms of a medical condition. Outside of this one letter switch to a different letter, there are other types of variants that we can find. In this example, the letter C has been completely deleted. In this example, the deletion is much larger, encompassing a sequence of letters. And in this example, it might be a little bit harder to see, but there's actually been an insertion of a letter. There's an extra C inserting itself into this sequence. And ultimately, all of these changes are impacting the integrity of that genetic instruction and carry the risk of changing its ability to do its job properly. For SLC13A5 deficiency, this is an autosomal recessive condition. So, taking you back to high school biology, remember that we have two copies of every gene. We inherit one copy of the gene from our mother and one copy from our father. So all of us on this phone call have two copies of the SLC13A5 gene. In an autosomal recessive disorder, each gene carries a genetic variant that impacts its function. So that's what I'm representing here with this little blue section of the DNA icon, is that each copy of the gene has a genetic variant that impacts the instruction coded by that gene. In most cases, when a child has an autosomal recessive disorder, we will find that both parents are carriers of that condition. So if we look here, you'll see each parent has one copy without the blue icon and another copy that does have that blue variant. So one copy of the gene is, quote unquote, healthy or normal, and the other copy of the gene is carrying a variant or a mutation. Carriers of a genetic disorder have that healthy backup copy of the gene, and as a result have no symptoms of that condition, and in this case would have no symptoms of SLC13A5 deficiency. As a result, you would never know you're a carrier of SLC13A5 deficiency because there are no signs or symptoms pointing to that. That said, as we know, each time a carrier of SLC13A5 deficiency or any other autosomal recessive disorder has a child, they will either pass on that healthy copy of the gene or they will pass on the copy of the gene that contains a genetic variant. It's a 50-50 chance. It's completely random. We have no control over what copy of that gene is passed on. When both parents are carriers of the same genetic condition, there is an increased chance that their child might be affected by that disorder. So for example, here we have two parents that are carriers of SLC13A5 deficiency. Again, they have that one pure black gene icon that has no variant, it's healthy. And then each parent also has a genetic variant that's represented by the blue. Each pregnancy, there is a 25% chance that each parent will only pass on the healthy copy of the gene, which you're seeing here. In this case, this child would have no symptoms of SLC13A5 deficiency, and they would also not be a carrier. So none of their children would be at an increased risk to have SLC13A5 deficiency. There is a 50% chance that one parent will pass on a healthy copy of the gene, and another parent will pass on a copy of the gene carrying a variant or a mutation. In this case here, I'm showing that the variant is coming from the father, but really it could be either mother or father. That's how we're getting this 50% number. If a child inherits only one copy of a genetic mutation or a genetic variant, they will essentially be the same as their parents. They'll have no symptoms of SLC13A5 deficiency, but that child will also be a carrier. Because many autosomal recessive disorders, including SLC13A5 deficiency, are so rare, it's unlikely that this child would go on to have an affected child. But it's sort of an important conversation to start thinking about as they get older so that it can inform their own family planning. And then the last scenario is that there is a 25% chance that each parent passes on a genetic variant in the SLC13A5 gene, which you're seeing here. In this case, that child would have SLC13A5 deficiency, and we would expect that they will develop symptoms of that condition. But one thing we always say is that it's hard for us in many cases to predict what exactly are those symptoms going to be and how severe will those symptoms be. We know that many genetic conditions exist on a spectrum, and we're still trying to learn what defines those spectrums. So just to recap, when both parents are carriers of SLC13A5 deficiency, every pregnancy will have that 1 in 4 chance or 25% chance to be affected by SLC13A5 deficiency. Some families may only have one affected child, and other families may have 2 or more affected children. So one question we get asked a lot is, 1 in 4 chance. If I have a child with SLC13A5 deficiency, does that mean I've hit my quota, I've hit my 25% chance, and none of my other children would be affected? Unfortunately, that is not the case. This is really an every pregnancy scenario, so every pregnancy is going to carry that 1 in 4 chance. When we think about autosomal recessive disorders in general, you'll find that when we test enough people, most people are going to be a carrier of something. So some of the bigger population studies that have been done by genetic testing labs suggest that between 1 in 6 to 1 in 4 people are carriers of an autosomal recessive disorder. And what's interesting is that it's actually possible to be a carrier of more than one condition. So for example, I'm a carrier of 2 conditions, my husband is a carrier of 4 conditions. It's really not uncommon to be carriers of a genetic condition. It's just that we all have no symptoms, so we don't really know unless we're actively being tested. One thing you may remember from your diagnosis is that in children who have an autosomal recessive condition, you might be asked if the parents are related by blood. And this is getting at a concept called consanguinity, which essentially just means that you share a common ancestor or you're related by blood. And the reason this question is asked is that individuals who are related by blood are more likely to be carriers of the same condition. So remember, I was just talking about, it's not uncommon to be a carrier of an autosomal recessive disorder, but what is a little bit more unusual is that 2 partners are carriers of the same condition. And so this is really where this question of consanguinity comes into play, because it might increase the likelihood that parents are carriers of the same disorder. And so I have a little diagram here to explain that further. Here I'm showing you the matriarch of the family who is a carrier. 1 healthy copy of the gene, 1 copy that carries a genetic variant. You can see that this woman goes on to have 4 children. And like we talked about, there's that 50-50 chance that she might pass on the healthy copy versus passing on the copy with the variant. So in this case, it actually played out perfectly. Half of her children carry the variant and the other half do not. We can imagine that 2 of those children go on to have kids of their own, and perhaps there is a marriage or a partnership between those children. So first cousins, which is really not uncommon in many parts of the world. By chance, we are seeing that both of those children happen to have inherited that same genetic variant, therefore meaning that they are both carriers of SLC13A5 deficiency. So this is really just pointing out, the only reason anyone ever asks this question is that it just increases the chance that people are carriers of the same condition and might change your genetic testing strategy in terms of diagnosis. After a diagnosis, this kind of information is important to know for family planning purposes because we know that children might be at risk, or sorry, I should say future pregnancies might be at risk for SLC13A5 deficiency or other autosomal recessive disorders. So it can just inform conversations around family planning and whether there are any reproductive options that the family might be interested in exploring, both before and after a pregnancy. So moving on, I wanted to talk a little bit about genetic testing reports. I'm assuming that many people on this phone call have at one point had a genetic testing report. Whether you were given a copy of that report, whether that report was walked through with you, whether the report made any sense is probably going to be variable person to person. So I wanted to spend a little bit of time thinking about that here. So this is an example of a report that Kim very kindly shared with us, and I'm through section by section. Even though this is a report from one lab only, and we know that labs are different across the board, every genetic test report is going to have the same information at some place on the report, and hopefully on the first page. So here on this is outlining is the gene or the genetic instruction where a specific variant was identified. So in this case here, we're seeing that there are two lines or two variants in the gene SLC13A5, and then one also in a gene called KCNH2. I find these reports are all so confusing because they're just a mixture of letters and numbers, but this is nicely labeled. We're looking at the genetic instruction specifically. The next column is really just showing us the coordinates of the genetic change. So where did that change happen and what was that change? The next column, we're seeing what we call zygosity, which is a fancy way of saying, did this change occur on only one copy of the gene, which is what heterozygous means, or is it actually present on both copies of the gene, in which case you would see the word homozygous here. And then lastly, every lab is going to offer an interpretation of those findings. And what this is really telling us is how confident is the lab that the variant they found could result in symptoms of a medical condition. Are they certain, where we would see terms like pathogenic? Or is there really insufficient evidence and we have an uncertain finding or an inconclusive finding? Breaking these down further, let's look at the very first variant that was found. Translating this gobbledygook into English, all this is telling us is that in the gene SLC13A5, the lab found a deletion of the entire sequence of that genetic instruction, meaning that all of the letters that make up the SLC13A5 instruction were not present. It was found to be a heterozygous change, meaning that this was present on only one copy of the gene, and the lab has interpreted this finding to be pathogenic, meaning that they're certain it impacts SLC13A5 function, which, in some ways you don't need to be a scientist to understand. If it's not there, it's probably not doing what it's supposed to be doing. And so here I just have a little sort of diagram of what that might look like. One copy of the gene appears to be normal in this setting. The other copy of the gene has a missing sequence or a deletion. So we would call that a gene deletion of SLC13A5. The second line is a little bit of a different variant. So what this one is telling us is that in the gene SLC13A5, again, they have found what we call a missense variant, where we see one letter being switched out for another. So the first section here, C.425C to T, is showing us the coordinates of the mutation in the genetic sequence or the DNA code itself. So it's telling us that at the 425th letter of that DNA genetic instruction, where we would expect to see a letter C, In this case, we're actually finding a letter T, a very, very small change. But if we look to the very next thing in the brackets, we find that this impacts the resulting protein sequence. So now what we're seeing is that in the protein, at the 142nd position, a T, or a threonine, is changed to an M, or a methionine. That's what those letters are standing for. So it's a little bit confusing, but remember, every DNA instruction codes for a protein. So they're basically describing the exact same variant, but giving us the coordinates in both the DNA and the protein sequence. That's why we see two things here. Again, this is a heterozygous change, meaning that it was found on only one copy of the SLC13A5 gene, and the lab is interpreting this to be likely pathogenic, meaning that they are very confident that this finding impacts the SLC13A5 function. So for us to put these two things together, what these results likely suggest is that from one parent, the child inherited a deletion of SLC13A5, and from the other parent, a child inherited that specific missense mutation. And as a result, both copies of the SLC13A5 gene carry a mutation impacting its function, and therefore this child likely has symptoms of SLC13A5 deficiency. You can imagine now, seeing how we're laying this out, why parental testing is often recommended. In the vast majority of cases, when a child has a recessive disorder, those variants are inherited from a parent, and testing is often recommended to confirm that. All right, I wanted to switch gears a little bit into reviewing some of the frequently asked questions, of course, in addition to any questions that you might have. So one question: how do I know if I'm a carrier? We talked about that in many cases, a child who has an autosomal recessive condition will have two parents who are carriers. But in genetics, we never say never. We're always surprised and we're always learning more. And so we do often recommend genetic testing for parents. So you may have already received this testing, and if not, you can certainly ask from it from one of your providers. Outside of SLC13A5 deficiency, there are also specialized genetic tests that look for carrier status across a number of more common genetic disorders, like things like cystic fibrosis, where people are more commonly carriers for those conditions. So those are also available to you if you're interested. And the reason why we even talk about these things is sometimes knowing your carrier status can inform conversations about family planning. So again, are you at risk to have a child with an autosomal recessive disorder in general, outside of SLC13A5 deficiency? And what are reproductive options that may be available or discussed at that time? Another question we get pretty commonly is whether the results of genetic testing are accurate. So what's interesting is that most labs actually use a second method to confirm the results of a genetic diagnosis, meaning that what you see on the report in most cases has actually been tested a second time using a completely different approach, because they really want to be certain that what they're reporting out is accurate. In some cases, the results from that first test are so clear and straightforward that they actually don't need to do the second confirmation. And so we're seeing that more and more frequently as our labs are getting better and better at doing this type of testing. And then another really common question we get is, what is a variant of uncertain significance? So these are actually really common, and they're going to be even more common in rare conditions like SLC13A5 deficiency. And essentially, we can just consider this to be an inconclusive finding. Not all genetic changes cause disease. So if you were to sequence everyone walking around, everyone is going to have genetic changes. It's part of just what makes us all unique and different from one another. And sometimes we just don't have enough evidence to know, is this finding just a normal part of who you are, making you unique? Or is this genetic finding disease causing? That is what this sort of uncertain middle ground means, where there's just not enough evidence to make that classification. But as we learn more over time, those results might change. So it's always important to keep an eye on our variants of uncertain significance. Many times they're reclassified to being a benign finding, so nothing to worry about. And in some cases, they are actually reclassified to be a likely pathogenic or a pathogenic finding, meaning that they could be disease causing. All right, and I think that is the end. So thank you guys very much. And of course, like I said, happy to answer any questions.
Thank you so much, Ellie. That was so great. I find genetics can be really challenging to explain, and I think you do it in such a clear way, especially with all the graphics. Okay, so
Oh,
I think
thank
what
you.
I'll do from here is I'm going to stop the recording so that people can feel free to ask the questions that are important to them. So just give me 1 second. And the portion that we did just record, we will send out to the larger group of families and extended family.